Awesome. Okay, so I got this recording and it's working. So, welcome to our Unit 2 review. If you watched last week's housekeeping, I did go through the major topics and concepts for each of the chapters within our particular Unit 2. If you haven't looked at that, please do. It's a longer housekeeping video because I talk about a little bit of like the general things, right? Homework assignments, exam, that sort of thing. But I also did take the time that I looked at the questions my seated students had, and I wanted to get something out to you guys so that you could work on studying that prior to the exam today. So I took those things that the seated students had questions on, and what they really like for me to do is just go through and highlight the main concepts from each chapter. And so I did that in the housekeeping last week. That's in Canvas, it's in the announcements. That video is posted out there. Please watch it because in that video, I also do talk about your cheat sheet. You are allowed to have a cheat sheet on this test. That is something that uh, a friend convinced me would be a good thing for you guys. And I'm going to try it. Okay. I'm going to trust that you guys are going to do what you're supposed to do. So I gave my seated students a square of colored paper. I can't give you guys a square of colored paper because I can't come to your houses, but I let them use both sides of a square of paper. This is just 1/4 of a piece of normal size paper. You can use one side of 1/2 sheet of paper. So you could, do that. You could cut one into a little piece if you want to, but you can do this. You can't use both sides. And you'll need to show me like this in the Respondus webcam video for the environmental check. And then I do want you to also send me an email with your cheat sheet picture. So send an email of cheat sheet, right? And this is one side of 1/2 page of paper. So basically you're getting the same thing as my seated students where they had a 1/4 of a sheet of paper both sides. You can use one side of 1/2 page of paper, and that's just the normal paper. It has to be handwritten. Right? No typing it up really small and then printing it and using that. Nope, 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 nope. Handwritten. The act of handwriting things out does help you remember it. So that's one of the reasons that I am doing that. In addition, no tables. This means if you are putting things out and you are listing them, products, reactants, by the steps, that's a table. Okay, that's still the table. I had a couple of students can draw, so a couple of students drew a picture of the um, like the chloroplast and wrote like the stages and then made a note to themselves about like the main like arrows of things going in and coming out. That's completely fine. So you can draw an image. You can draw the flow chart. Um, you have to be able to convert that information into a table. So I don't want you to copy paste ever in my class, and having the table handwritten out on a piece of paper counts as copy pasting because you don't have to actually know it, and then that ends up affecting your ability to do the rest of the exam. Just like with the biomolecules table, the reason I'm making you memorize this is that you can understand like where we use the oxygen, where we get the water from, where we're using water in photosynthesis, and then how these things all work together to give us a full comprehensive understanding of energy processing and why that's so important. So make your cheat sheet. Okay, send me a picture of it. Be honest. I trust that you guys are going to be honest and have integrity with this. If I have questions about it, you could end up getting a 0 on the test because if I question your integrity, it's a violation of academic integrity and becomes a dishonesty issue. That becomes a problem. So don't make me regret it, okay? If you don't make me regret using a cheat sheet, I might let you use them on future tests. But that is something that I think would be helpful for you guys because just having the act of making a cheat sheet is a fantastic way of study, which brings me to my next topic here. Um, and what's happening here is a couple of people asked me how to study, how to learn this stuff, how to study. Well, one, you need to just learn it, okay? In an online class, in a seated class, it doesn't matter. When you're in a biology class, and I know I've said this before, it's a new language, okay? It's some bigger concepts. It's things that maybe we haven't thought about in depth and detail, and we're looking for connections. And so a lot of it is supporting your reasoning. A lot of it is puzzling things out, and then putting those together into a whole big picture. So part of the way that you're going to have to work through that is by reading the textbook, by doing your homework, by doing your quizzes. You can go back through the quizzes, and sometimes I do pull questions, not the multiple choice. Those are just to like check your understanding, okay? But any of those short answer quiz questions are free game for the test. The matching, the fill in the blank, the short answer. I do like to pull those onto the test. Some of you probably realized that last time, because why would I reinvent the wheel, right? Those are the things I want you to know. That's why I'm asking you on the quizzes. Those are the things I want you to know. You're going to see a similar version of that on the exam then. Make sure that when you're writing your cheat sheet, you're thinking about the things that you can't easily remember, and you're making it in such a way that it's shorthand. Having a cheat sheet that is just chock full of words that you've copied down directly from a source is not helpful to you, okay? It should be as you're studying and you're making kind of notes to yourself, kind of like the bullet points that I did on that how to, what to do for Unit 2 home housekeeping video. That's the sort of thing that you should be putting on a cheat sheet. It shouldn't be all of the details, but if you're like, man, I can't remember competitive and non-competitive inhibition, draw a picture of them. Because if you see an image, you'll remember what they mean. And the image is easier to interpret than some of our own handwriting. Okay, and I'm saying that after reading some people's handwriting. In addition, I'm a big, big, big fan of flashcards, and flashcards will become even more important in the next unit. You can use the ones that are pre-made in Quizlet, just make sure that they're correct. Or McGraw-Hill has an app called Sharpen, SHARPEN, that is free to you as a student who's signed into a class, and they have flashcards made for you on the vocab. And since everything is from the book, that would be a good way to make those flashcards and study them. Your objectives are the things you need to know. Those are the things you need to learn. Those are the things you need to study. So I do talk to those in the lecture videos. In addition, so as I'm going through the lecture, I'm keeping in mind what those objectives are to make sure that I'm hitting on them. But in addition, those are the things that you should be able to answer. If you can answer all of those as questions. So if you look at it and it says, compare and contrast photosynthesis and cellular respiration. That might be a question that's like, what is the relationship of cellular respiration and photosynthesis? And you should be able to say that the products of one are the reactants of the other. So that's what you might see for those objectives. You can pop those into ChatGPT and ask it to write you questions. That is a completely acceptable way to use an AI system. Okay, your check your understanding questions are built off of the objectives. Those are the ones that I have in the PowerPoints, and I have answers in the PowerPoints in the notes section. So also that would be a place to go and study from. And then yes, last week's housekeeping video, I went through and I did the kind of breakdown of each chapter. What are the main concepts? What do you need to know? Why do you need to know them? And so I know that's a little bit longer, but that's because that's what that's for, okay? So today we're going to go through those main things again, and I'm going to do a little bit of detail on those. If you have questions on any of this in particular, let me know. Most of you, I looked through the Chapter 7 attendance quizzes and all the other attendance quizzes, and you have questions that are like expansion questions, which are fantastic. But some of you had some questions on particulars. So let's talk real quick about ATP. Remember, ATP is cellular energy. Solar energy is sunlight that comes into plants. Plants convert solar energy into cellular energy. They convert it to ATP first, and then that cellular energy is converted to chemical energy, which is stored as glucose or starch. So what happens here when we're talking about ATP? ATP is not stable. Okay, ATP is a cycle and it breaks consistently. It's being made consistently, it's breaking consistently. So it's not able to be used as long-term energy storage. So what we actually do is we have solar energy that is converted into ATP, which is cellular energy. which is then converted into glucose or starch for long-term storage as chemical energy. That chemical energy can then be used in cellular respiration and go through the process of releasing ATP again. So basically, we're kind of like putting ATP into a storage and then able to use it again from that source. So remember that carbohydrates are carbons, hydrogens, and oxygens. This is why plants need carbon dioxide and water. And this is why when you do cellular respiration, you release carbon dioxide and water because we're breaking down that carbohydrate. And at the end of the day, it's all about the energy. Where are we getting our energy from? Those carbohydrates or lipids or proteins or anything, but your cells prefer to use carbohydrates as the primary source. Okay. When we talk about enzymes, okay, they're not used up. They actually facilitate reactions. So they lower the activation energy required for reactions to occur. As such, when you see a figure, okay, like this graph, and the graph says amount of energy is increasing on one side and reaction on the bottom side, or how, we're getting to that reaction, baby, at the end, we can compare a reaction with and without an enzyme. Without an enzyme is going to require a lot more energy. Remember, this is like building a bird box with a hammer and nails. You can do it, okay? You start with the same reactants, you end with the same products, but it takes energy to pound that hammer onto those nails and get the nails into the wood. Whereas if I want to build a bird box more efficiently and more quickly, I'm going to use a nail gun because the nail gun lowers the amount of energy I have to put into that reaction. So when we talk about things like glycolysis, okay, there are totally enzymes involved in that. Your cells don't just randomly break glucose. There are proteins that notice glucose and they actually connect to the glucose at the active site and break that sugar. So that is going to be part of that process there. Okay, we take a little energy to do that because it takes energy to break glucose. We also need to remember that enzymes have an active site, okay, that substrates can bind to, and they are substrate specific. So an enzyme is only really capable of binding to one substrate. And when it binds to that substrate appropriately, it's the lock and key, where if you put the correct key in the door, right, in the lock, it will allow you to turn the lock and unlock the door. You might be able to fit a different key into the lock, but it's not going to turn. And so when we talk about the lock and key, that's what we're saying. Enzymes are substrate specific. And this means that they do not act on all products or all substrates. When you see something in science, if you ever see anything that says all or all or always or never, you should be a little bit skeptical of that because generally speaking, that's not the case. There is exceptions to the rules for almost everything. And so we're going to see that within things like enzymes as well. But generally speaking, substrates are specific to particular enzymes and vice versa. We can stop enzymes from functioning by having things like competitive inhibitors that block the active site or non-competitive inhibitors that change the shape of the active site and don't allow it to function. The Amoeba sisters made a joke about like, the if I fits, I sits. If you have cats or ever seen cats, right? If they fit, they sit. Doesn't matter if they like barely fit. And it's kind of funny because that's what happens with competitive inhibition. That enzyme cannot bind to the substrate, and that's why that substrate is like, hey, that's my enzyme. Um, because, oh, hi. Because this enzyme cannot bind here anymore. Okay, cool. Josiah, if you have any particular questions, just pop them in for me. I just started kind of going through the basics here and recording, so I'm going to keep going. But if you have any particular questions, let me know, okay? All right. So enzymes, okay, remember substrate specific, okay? You will probably see this figure in some way, shape, or form on the test. Make sure you understand it and make sure you understand the difference between competitive and non-competitive inhibition. When we go through chapter 5, then, after ATP and enzymes, we need to talk about cell transport, or more specifically, diffusion and osmosis. You might see questions like active transport, and that requires energy, or bulk transport, where it's for large molecules. But generally speaking, we're concerned with diffusion and osmosis because that plays a role in cellular respiration and photosynthesis with those hydrogen ions and that electron transport chain. So diffusion is a movement of solutes and solvents across a membrane. So this is a movement of salts and waters across the membrane from high to low concentrations. And generally speaking, okay, we're going to see that for mostly everything, as long as it can move across the membrane. Remember, not all membranes are allowing everything to move across them. Some membranes are not permeable to certain items, including large things like glucose or even small things like ions. Your membranes are your chain link fence, right? We want to keep our dogs in the yard, but we want to allow the birds, like the little wrens, to move through the fence unimpeded, or the insects or the lizards or whoever it is. We don't want strange dogs coming in. So, we're talking about what your cells are allowing in and out across the membrane. Osmosis is a type of diffusion that is specifically a solvent, which is typically water, because remember, water is our universal solvent. We learned that in chapter 2. And generally, this is going to happen when the solutes can't move. So salts are not able to pass the membrane, which is good. Most cells keep their salts. And so water moves from areas of high to low water concentrations, which generally means waters follow the salts. So if we look at cells in different osmotic environments, okay, we have let me draw a little text box here, okay? We have isotonic, hypotonic, and hypertonic solutions, okay? Um, when we look at these solutions here, okay, we need to determine which direction the water is moving. So I like to put in the little math question because it helps me make sense of things. So if my normal cell, right, is 70% water and 30% salt. Okay. Now, is that accurate? No. Can I make that if I want to? Sure. I can make a pretend cell with that concentration. Okay. An isotonic solution will also be 70% water and 30% NaCl. Let me admit. Boop. Okay. Um, when we have this isotonic solution, then it's going to be the same as the red blood cells in this case, because red blood cells, animal cells, they just have, it's like a balloon, right? And if I overfill a balloon, it will pop. If I underfill a balloon, it looks all shrivelly and shrunk up, like if I fill it and then like release air. But if I just fill the balloon and it's happy and balanced, I'm going to see that like isotonic sort of situation. Now, animal cells like to be in an isotonic solution. So animal cells are really happy if we have isotonic solution. Plant cells wilt. So they're not unhappy, but those are your dramatic plants that just kind of fall over and are droopy and sad if they're in an isotonic solution. They need more water than what we need in our animal cells. So when we see animal cells in an isotonic solution, they will always look normal, especially if we're looking at like red blood cells. This would be like if someone, oh, you'll have a hypothetical question on this test, by the way, where I'll ask you in a hypothetical situation if the solution provided matched the cell or not. So it could be something like if you put a saltwater fish, what you thought was maybe a freshwater fish, into fresh water and it died because water moved into the cells, what kind of solution was that? Or I might ask if a doctor thought they gave a patient an isotonic solution and the patient died and we saw blood clots, what kind of solution was probably given to the patient and why. And so I need you to kind of think through that process there. So let's talk about what would happen then. Well, in a hypotonic solution, the solute and solvent concentration is different. So if we put a cell into a solution that is hypotonic, there's more water in the solution than there is in the cell. So this would be like if I did a 90% water and a 10% salt solution. Okay. In this case, there's more water moving into the cell than it can handle. So water moves into cell. Okay. Animal cells burst. Plant cells are happy because plants have that cell wall. Plant cells have that large central vacuole, and that provides them with that physical structure and support. And this is what plants actually use in a lot of cases to combat gravity. So for plants to be able to stand upright and not be tipping over to either side, they need to have those cells nice and full of what's called turgor pressure. And so it's just this pressure on those cells. And we can see that if 90% water, there's more water than we see in the cell by 20%. 20% of that water is going to try to move into the cell in order to get it balanced, or even 10% of that water. Whatever it is, it's like overfilling the balloon. If I overfill the balloon is going to pop. And that's going to be the case. That would be like what would happen if you took a saltwater fish and you put a saltwater fish in a freshwater environment. Water moves into those fish's gills faster than it can handle it, and it actually causes the cells in the gills to lyse, and that causes the fish to drown because it can't get enough oxygen to do the gas exchange to get ATP. And so that's that connection there with this process. A hypertonic solution isn't going to be good for any cells. Hypertonic solution would be if I had 50% water and then 50% salt, right? In either case, right, I have less water than I would have in my cell, okay? And so this is going to result in dehydration. So water is going to move out of the cell and no cells are happy, okay? Animal cells shrivel and clot. Plant cells wilt and die. We call that plasmolysis in plant cells because actually the cell membrane pulls away from the cell wall, which is not going to be good for the cell. But generally speaking, this is what we see in these osmotic environments. So you will absolutely have questions about those, as well as a comparison of diffusion and osmosis. Okay. Questions on that? If anything, as always, just send me a message, let me know. And then we have now finished a lot of the main topics for chapter 5, okay? Don't forget your cheat sheet. You can draw these images. I would draw pictures of the cells, personally. I would draw the picture of the cell and put the little math things there to help me, or show arrows of water moving in and out. Just a thought. Just a thought. Okay, so that covers chapter 5, all of those main concepts there. Vocab is fair game for everything, so just remember that. And now we get into photosynthesis. So one of the things that people get stuck on is when we're looking at things that are really small, beyond microscopic, because these are in organelles and we're in the membranes of the organelles, and I can't effectively show you these processes, which is really hard. This was always a hard thing for me to grasp as a student as well. I am a whole organism, whole ecosystem person. You want to talk about how carbon is moving in and out of organisms? I got you. But when I was a student, like, this was hard for me. And part of the reason is because I can't show it to you. I can't take you into a cell and show it to you. I wish I could. I wish someone would buy me a magic school bus and we could go on adventures and field trips. But what we do know is that plants are essential to our ecosystem. Okay, without plants, we cannot convert solar energy into that cellular energy, which is then stored as that chemical energy. And so we absolutely need plants. Now, do plants need us? Not really. Plants can do their own photosynthesis, and they can actually do their own cellular respiration because they do cellular respiration as well. Remember, plants do both, Animals only do cellular respiration. And as such, plants can go back and forth in this equation on their own. Okay. But we're going to fill out the steps. We're going to fill out a table. Okay. You can put some version of this on your cheat sheet, but not a direct table and not something labeled like a table. I would suggest drawing a diagram and having that diagram be available to you to help you understand, especially because diagrams show us the connections to things. You will have a table on your test. Study the tables, but do not put it on your cheat sheet. That is not allowed. Okay. So when we look at photosynthesis, our equation is this understanding of carbon dioxide and water and sunlight make glucose and oxygen. And we're always just rearranging these chemical bonds. And in rearranging chemical bonds, we get different energy. We can do different things with the elements as we release them. And we can make new products because these are like the Legos, okay? Energy is lost eventually. And so sunlight is coming into the ecosystem and is used to jumpstart that process. It's like turning the ignition in your car and starting the energy process. Okay, so if we look at the equation itself, I would strongly recommend writing the equations on your cheat sheet because just having that is enough to help you fill out the table. But we start with 6 carbon dioxide, 6 water, plus energy, not 6 energy, just energy in general. And we get out glucose, which is C6H12O6 and 6 oxygen. So we know that somewhere in this table, our outs are going to include glucose and oxygen. And if I ask myself, what is the very end product of photosynthesis? What is my end goal of photosynthesis? It's glucose. So my very last box should be glucose, which is that C6H12O6. Okay, that is my end goal. So I have to ask myself, how am I getting the carbons, hydrogens, and oxygens to build this carbohydrate? Okay, well, this is where things might get a little tricky, but we know some things. We know that the first step is called the light dependent reaction. Okay, light dependent reactions require sunlight. Light dependent. Okay, my second step is the light independent reaction, and it doesn't require sunlight, also known as the Calvin cycle. I just prefer light independent reaction because it already tells me something that I need to know. The light dependent reactions are going to occur when it's sunny. So when it's sunny, which also tells me then that my in here has to be sunlight. I have to include sunlight as the start of this light dependent reaction. Again, that's like the sunlight is like turning the key in your ignition. It starts that process. Okay, my light independent reactions occur 24/7. These are slow processes because they're making something. It takes longer to make something than to break something. So if I were to put something on a cheat sheet, I might put something like this breaks water, and I might put something like this makes glucose. Now, this is telling me enough that I should be able to, with the equation, fill out the rest of the table if I know that kind of process there. Why does that make sense? Well, because if my light dependent reaction is breaking water, okay, I know that water is H2O. I also know that that has to be an in here then, right? I have to have water going into this process. If I am breaking water, I am getting out hydrogens and oxygens, because if I split water molecules, I get out oxygen and I get out hydrogens. Well, if I look at my equation, okay, one of my out products is oxygen. So I know that this is where I am getting my oxygen out based on my equation, because I'm not getting glucose out of the first step. If I got glucose from the first step, I wouldn't do the second step. And I know that I've split water. And so oxygens are going to be a byproduct of breaking that water molecule. But, okay, I need those hydrogens if I'm going to build glucose, because glucose is C6H12O6. And I have 12 hydrogens in the ends of my reaction. So what I need to do is trap those hydrogens, and I do that on a molecule called NADPH. This is a coenzyme, and it transports those hydrogen molecules. It just picks them up and moves them. But I know from my ATP cycle, or from this idea of solar energy to cellular energy to chemical energy, that I have identified my solar energy and I have identified my chemical energy of glucose, but I am missing my cellular energy. And if I want to make chemical energy from solar energy, I need ATP. So ATP has to be one of the products of the light-dependent reaction. Oxygen is a waste product, but I know that these two, NADPH and ATP, have to go into building the glucose. Okay, they have to go into building the glucose. And if I look, I have my hydrogens here, but I do not have my carbons or my oxygens, but plants need carbon dioxide for that purpose. And so my carbon dioxide is going to go in, okay, to this step. And I could probably change these colors. These are going in. Let's change those to red here, to this step. And as a result, I have now gotten all of the things that I need to build a glucose molecule. I have the energy, I have the carbon dioxide, and I have the hydrogens. So I have carbon, hydrogen, oxygen, energy. I can now rearrange those molecules and make glucose. But it is a slow process. And so that's why this runs 24/7. If I am breaking water, that means I'm doing an electron transport chain. That tells me this has to be a thylakoid membrane. And if I am doing a cycle, I need space, and cycle is going to occur in the stroma because it needs the space in order to build that product. You will need to know this for the exam. Okay. But again, it's not always just strict memorization. It's talking through it. So say the things out loud as you're working through studying them. I strongly recommend that. Say it to a plant, say it to the wall, say it to a kid, say it to a spouse, say it to somebody or something, because the act of talking through it and the act of talking through it with a puzzling out of where things belong, like I just did, is what reinforces it in your memory. When I first taught this, it was terrible. I was terrible at this. The more that I've gone through it, the better I can explain where my logic is, the more I can support my reasoning, the more I can use the vocab words, the better off I am. And that's going to be the case for Unit 3 as well. Unit 3 is very vocab heavy, so be prepared to start using those words regularly. Just a quick reminder of what the light-dependent reactions do is they trap solar energy. You have chlorophylls A and B, which trap the blue-violet light and reflect green light. And you have carotenoids, which trap the green light and reflect orange. And when that light is caught by those chlorophylls in these embedded membrane proteins, this is how I am going to break that water. I'm using the energy from the sun to take the hydrogens off of the oxygen. Oxygen always holds onto the electron tighter because it's a polar covalent molecule. So when I pop those hydrogens off, the electron is left behind. These proteins are specialized to be able to pick up that electron, and that electron is then used as an energy source as it's being passed from one protein to another to move hydrogen ions from an area of low concentration to an area of high concentration. So we're moving across the concentration gradient, and we are building up a large amount of hydrogen ions. And so we are creating a situation where, one, we have acidic solution because lots of hydrogen ions are acidic, and Mother Nature hates concentration gradients. We just talked about that with diffusion and osmosis and how it wants to equalize those. So these hydrogen ions are looking for a way out. They cannot pass the membrane. The membrane is not permeable to them. The only way they can go out is through the gate, which is ATP synthase. So as they move through that enzyme, ATP synthase is an enzyme that makes ATP, and it uses those hydrogen ions. So we're using hydrogen for two purposes in these reactions. One, to make the ATP. Two, to make the glucose. So these hydrogens are kind of dual purpose in this process. Okay? We're going to go through the table for cellular respiration as well. And fermentation, I just wanted to make a note. Fermentation is an alternate pathway of what happens when cells are in anaerobic conditions. In the lecture attendance quiz for chapter 7, several people asked me what would happen either one, if cells didn't have oxygen, 2, do we have a slight store of oxygen and what happens if we run out? 3, what do small cells do when there's not enough oxygen? How do anaerobic bacteria work? There were a lot of questions about anaerobic processes. Anaerobic, remember, means without oxygen. So our cells do have an emergency mode called fermentation. And when you are actively working out, your cells have a store of oxygen. Okay. It takes a little bit of time from a single breath for that oxygen to diffuse from your lungs into your bloodstream and then move around your whole body and diffuse into the cells that need it. Okay. So we have, because you're breathing constantly, a buildup or a store of oxygen. If you hold your breath for a long time, you're going to start getting a buildup of carbon dioxide in your blood, which is how your body knows that there's a problem. So that buildup of carbon dioxide is going to trigger that homeostasis is not good in your blood because CO2 is acidic, and you'll start getting that like signal that you have to take a breath. And sometimes you can't overcome it, right? If you hold your breath for too long and if you pass out, your body will just restart those processes again. So there is a store of oxygen in our cells, which is why things like Olympic sprinters don't breathe while they're running the 100-meter dash, because they have enough oxygen in their cells to get through that process, and breathing is just going to slow them down. They can just hold their breath for that, I don't know, what, 3 seconds or something that they run that in, and be fine. So we do have fermentation as a pathway, and it is a viable pathway instead of having any sort of cellular respiration and requirement for oxygen for a lot of microorganisms. They don't need that much ATP, or they are effective at making ATP without oxygen. They have other electron acceptors like sulfur is a big one. So you get hydrogen sulfide as a result. There's a lot of different ways that that can happen. Okay. In our cells, we talk about fermentation building up lactic acid because our body uses the lactic acid back into pyruvate. In fermented foods, that tanginess of fermented foods is also a byproduct of fermentation. And a lot of times that's kind of a lactic acid sort of buildup. Okay. Again, though, if we look at our equation for cellular respiration, I would put that on my cheat sheet. Okay, I would put it on my cheat sheet, and I would say that I know it's the opposite of photosynthesis. And so I'm going to do the same thing with filling out this table. I'm not necessarily going to go in a straight order because I'm going to fill out what I know effectively and help me puzzle out the rest. So I would always start by knowing the steps. Okay, the steps of cellular respiration are a big deal. You need to know that there's glycolysis, which is the breaking of glucose, glycolysis. Okay, we need to know that there's a why is that small? Let's turn that up to 16 here. There's a citric acid cycle, also known as the Krebs cycle. And again, I prefer citric acid cycle because it's different. Okay. And then there is the electron transport chain. Now, I'm going to put a note here about fermentation, but this is not needed on the, not included on the table on the test. So it's not on the table on the test, but people have questions about it. So I wanted to talk to it. Okay. I skipped this one for now. We're going to come back to it in a second here. What I would personally do is if I was writing a cheat sheet for this, I would draw a mitochondria and a cell, and I would label the steps, what they are, and I would probably just abbreviate them. I would put the G outside of the mitochondria, the CAC in the middle of the mitochondria, and the ETC along the membrane of the mitochondria. Again, I'm not putting a table on the test, but I'm showing myself where those things go. I know glycolysis occurs in the cytoplasm. And this makes sense because what I'm doing here is glucose is a large molecule, okay? I cannot take glucose directly into the mitochondria. I have to break it into itty bitty pieces and feed it to my little pet mitochondria. Okay, we breathe for our mitochondria. We have glucose for our mitochondria. We love our mitochondria just like we love our pets. And so for me, that helps me make sense of this. Plus, I know that glycolysis Lysis means to break, and glyco is a sugar. So knowing that the first step is called glycolysis, I know that I'm going to start with C6H12O6. I have to start with glucose. I can't do this process without starting with the sugar molecule. Okay, I need that glucose to start this process. I am going to skip around a little bit because, again, for me, it's about the logical progression of things. The citric acid cycle I know occurs in the mitochondria, and the electron transport chain I know occurs in the mitochondria. And like I said, I would draw a picture and I would put that this is in the matrix in the middle, and this is in the mitochondrial membrane, also known as the cristae. If you put mitochondrial membrane, that's okay. Cristae is the more accurate term, but that's fine, okay? If I'm in the mitochondria, okay, I'm looking at my equation here, right? I know where glucose is going in, but I have oxygen that has to go in as well. I breathe oxygen for my mitochondria to produce energy. I breathe oxygen, therefore, for my mitochondria. If I am anywhere in the mitochondria as my location, I have to put oxygen into that process. The end, period. So I've already determined where all of my equation reactants are going into this process. Now, obviously, this isn't enough because if I'm making something in step 1, I'm probably going to use parts of that at least in step 2 and then in step 3 because I wouldn't do these as individual standalone steps. I am doing something so that I can go from step 1 to step 2 to step 3. I am not just skipping around in that way. These are a logical progression within the cells and within the organelles. And so glycolysis in the cytoplasm is going to take in that glucose and it's going to do something with it. In this case, I need a little bit of energy because if I want to break a big item, like if I wanted to break this pen, I have to put a little oomph into that. So I have to put a little energy into breaking my sugar molecule. So I use a little bit of energy here, okay? And that gives me 2-pyruvate, which is a 3-carbon sugar. Well, C6, right? So I have 6 carbons here. I still have 6 carbons here. And so that 2-pyruvate, okay, is going to also give me 2-NADH because I'm popping the hydrogens off of my glucose molecule, and I'm going to get 2-ATP. Now, my 2-ATP, anytime I see 2-ATP on this side, I'm done with it. It's good to go. I'm going to use that ATP, and I'm just done. I don't have to think anything else about that, okay? Whoop. I look at this pyruvate, though, and I go, okay, well, that's still a sugar molecule. It's still a 3-carbon sugar molecule. I don't have all the energy I can get from it then, right? I need to be able to break that further. So my 2-pyruvate is going to go into my next step because if I made a 3-carbon sugar so it can move into the mitochondria, I'm going to see it moving into the next step of the process. So I now have oxygen and 2-pyruvate. Okay, my mitochondria can do a lot with this. It's going to take this process and it's going to slowly break this pyruvate into its individual carbon molecules. The big end goal of both glycolysis and the citric acid cycle is to make the carbon molecules like off the glucose, take the carbon molecules off the glucose, but the really, really, really big end goal is to trap those hydrogen ions because once again, we have ATP synthase playing a role. And I would put a note here that this is ATP synthase, which means I need hydrogen ions. So what I am doing here is I am taking a carbohydrate and I am trying to capture all of the hydrogen ions possible. I do that by removing the carbons and breaking that molecule into its component parts. But I don't want loose carbons. That's why I have the oxygen, and that ends up making me CO2, which is one of my products, CO2. I've identified it. I'm done with my CO2s. I've got it. We're good. Okay, so that takes care of that part of the equation. I have my oxygen and my glucose identified. I get a little bit of energy here too. I don't get a lot of energy. I get a little bit of energy. It's not very much. Because again, if I got all of my energy from step 2 from the citric acid cycle, I wouldn't even do the electron transport chain. I wouldn't even go to that last step. In addition, okay, my carbohydrate, my pyruvate, I am going to strip off all of those hydrogens. So here's the big players. I have 6 NADH and 2 FADH2. These, I'm going to change color here again. Let's turn those, I don't know, orange, an orangey color. And my NADH here, I'm going to turn that an orangey color too, okay? Those are my hydrogens. I have 6 times 1 is 6, 2 times 2 is 4, so I have 10 hydrogens here, and I have 2 hydrogens here for a total of H12, or 12 hydrogens. These hydrogens are going to be my big players in this next process. So here I'm going to put in all of my hydrogens, all of these NADHs. These NADHs and FADH2s are Ubers. They are picking up the hydrogens, they are trapping them, and they are moving them where we need them to go, in this case to that ATP synthase. Once again, we're going to do an electron transport chain. We're going to build up a concentration gradient. Mother Nature hates that. Those hydrogens, the only way they can escape is by moving through ATP synthase. They're going to do that, and they are going to generate a ton of ATP. At this point, right, this is where I'm going to see the most of my ATP being made. And because of that, I can be done with this process. Hooray! But I haven't identified all of the parts of my equation. In this case, I am still missing my water out. Well, I don't want oxygens and hydrogens just loose, okay? Hydrogen ions just loose are not good. And so in order to make those into a product that can just be released, a product that is not going to hurt anything, I'm going to make water molecules. Okay. And this also illustrates how photosynthesis and cellular respiration are opposites of each other. In photosynthesis, we start with an electron transport chain that splits water. In cellular respiration, we end with an electron transport chain that makes water. But in both cases, it's still the same enzyme and it's still the same process where we are creating this concentration gradient, and these hydrogens have to go someplace, and the only place they can move through the membrane is through that enzyme that makes ATP, okay? If I come back here though, I did leave a thing blank here. I want to make a note that this is step 1.5. This is extra credit. If you happen to know this on the test, great. This is your preparatory reaction, also known as what I like to call the prep step because it rhymes, okay? This occurs in the mitochondria, okay? And in this, we put in 2 pyruvate, and we put in a little bit of oxygen because we're in the mitochondria. It has to have oxygen. And we get out something called 2-acetyl-CoA, and we get out like NADH and a couple of other things. Honestly, okay, if you know that much, that would be extra credit. The pyruvate then wouldn't be what goes into the citric acid cycle. It would be those acetyl-CoA. Acetyl-CoA is a 2-carbon sugar, and so we're taking off another carbon. So technically here we'd have 2 CO2, and then this would be 4 CO2 if we happen to know that. Again, I'm not overly concerned about that. That's just kind of extra credit. If you happen to know some of that, I'll give you some extra points in the test. I need you to know glycolysis, the citric acid cycle, and the electron transport chain. Fermentation is not included because fermentation also is in the cytoplasm, and this is a alternate pathway when oxygen is not available. So it's an anaerobic process. In, we would put in 2 pyruvate, out we would get lactic acid and ethanol. And 2 ATP. That's it. We get 2 ATP from this process. So it's not the best process, but it is something that can be used short term as an emergency source of energy for us, or long term for a lot of small organisms. 4 ATP is a lot of ATP for small organisms, and they do this so much faster than our cells do. Okay. There's no blood movement, moving things, diffusion across cell membranes. Like, they are just pulling in sugars and just going to town. So we do see them being way more effective at that than we are, okay? Last but not least, this is that kind of from that video of the pre-lecture for the flowcharts, just showing us again the relationship here. These two processes are opposites of each other. The products of one are the reactants of the other. They are very intimately linked. They maintain the carbon system in our atmosphere. They keep oxygen moving around. They keep energy available to our systems. And obviously, we need both of them. Plants need both. Plants cannot grow without cellular respiration. Plants can make their own energy, but once they store that energy, they have to be able to break it down again the next year to grow. Um, we rely on respiration, right? We know that we have to eat every day. So that's what we have as that. That's covering everything in Unit 2. Okay. Josiah is typing. Let's see if he has a question here. Yeah, I'm gonna download it and get it uploaded as soon as I can. Today it depends on how fast it downloads. So, um, I went through ATP and this idea that solar energy is cellular energy to chemical energy from photosynthesis, and then enzymes, and then you popped in for the rest of it. So you didn't miss too much as far as reviewing the materials. I did talk about the cheat sheet, so you can use one half of a piece of paper and just one side handwritten. So you will have to send an email of that and show it in your respondus um, for that, but that's really all you really missed. So I will get this downloaded as soon as possible and upload it into Canvas. I have to like download it and then upload it to YouTube and then share it. It's a little bit of a process, and I have class at 9:30, so I'm hoping I can get it done before then. If not, it will be up before noon because I have class again at noon and I have some time between 11 and 12. So I will have this up by noon today. The exam opens at 11:59 p.m. tonight and closes at 11:59 p.m. tomorrow, the 24th. So you have a little bit of time to study. Sharing. Cool. Let me actually, the sooner I think I tell it to stop recording, the sooner it starts